저도 사장님이 너무 얼굴이 밑에
Good morning.
Good morning. Hi, nice to meet you.
Nice to meet you too.
Yeah.
Sorry
First,
for sorry for being late. I have problem with my internet this morning. I hope it's gonna be okay.
yeah, okay. Can you hear me very well? Yeah, okay, perfect.
All
Yeah,
good.
first let me introduce our team. Yeah,
Mm-hmm.
we are mainly in charge of this about some application support and trade related things. And my name is Austin, and his name Nathan.
Mm-hmm.
And this is Carlos. Yeah.
Nice to meet you all.
Bonjour.
Yeah. First, we are very appreciating you taking time to arrange this training course. Yeah.
My pleasure is shared, and thank you so much for your time as well.
Yeah, you can start.
So I already have two options for you here, guys. So because I know you've uh done your homework and you've studied well all the elements that I shared with you, we can really do it two ways. Either I can give you the introduction, but that might be a little boring and annoying, or as you've been through the different elements that I shared with you already, I can answer your questions. What do you think is best, and how do you want to proceed?
Okay. So
Tell me which.
do you
Go
have
ahead.
some PowerPoint or something you have to share?
Sure. Sure, I can definitely do that, but I just want to make sure it's the most useful to you, and you maybe have already had a look at them, so it's really all up to you. I can
Mhm.
either answer your question or give you just the introduction. Okay, I'll pull out the slides, and then we'll try to go from there on. Okay?
Yeah.
So it's sorry, as I said, my internet is a little bit weird this morning, so everything is a little slow on my side, but it should be all right.
Yeah, it's fine. Taking time.
All right. Can you guys see my screen?
Yes, yes. I
Sounds
can
good.
see clearly.
Okay, so if I speak too fast, or if you have any questions at any time, you feel free to interrupt me, okay?
Okay, thank you.
Awesome. So very quickly, so Insight Therapeutics, we're a company of we are a bit more than 20 people now, based between Paris and Bordeaux in France, all right? We have a bit more than 50 customers worldwide, and I'll come back to you on this. And we have several patents on nanoparticle formulation, right? Our core expertise really is making nanoparticle formulation equipment. So this is really what we aim at doing. And just to tell you a few words about where we come from. So originally, three of our co-founders, including me, we were working for Elveflow. Elveflow is this French company that is one of the world leader in microfluidics. I think you know the field of microfluidics. And basically, what happened is that when the COVID vaccine came out, many people reached out to us looking to use microfluidics for nanoparticle formulation, for LNP, RNA LNP formulation mostly. And basically, what we realized back then is that there was only one company making such kind of equipment. They were Precision Nanosystems, and they were bad, and at the same time, they were very, very expensive. So we were like, okay, there is a lot we can do to help. In parallel to this, we also were providing homemade systems, and we very easily understood that they were way too complex for the end users. So eventually, what we did is that we took those two elements, so these technical requirements and this request from users to start Insight Therapeutics in 2022, really with the goal of trying to bring all our microfluidic technical knowledge to the RNA lipid nanoparticle formulation. We have several patents also on a unique technology, but this is what is going to happen next year and the year after. I'm not going to talk too much about it today, but basically at the moment, we are just using a regular microfluidic technology. Make sense to you? All right. So to start with, a quick reminder on RNA therapy. So just to start with, so we mentioned a lot all the RNA, all the nanoparticles, and we can pretty much cover any organic type of nanoparticles, and I'll come back to this later. But I would say 80 to 90% of our users, they work with RNA therapies or RNA vaccines, okay, for RNA lipid nanoparticles. So this is why I will focus on this, and then I will open up a bit more. So a quick reminder, so what is this RNA therapy? These RNA therapies are basically new type of therapy that people expect to be like the next big thing. And basically they use, the goal here is to directly bring into the cell this RNA, so this ribonucleic acid, within the cell. Okay, maybe let me ask you this question, guys. What is your background? Do you have more of a physics, chemistry, biology background? What's your scientific background here? You guys on your side, what is your scientific background, if I may?
Biotechnologies.
Biotechnologies? Okay.
And personally, I am microfluidician, so yeah, I'm well know about the technologies involved in this machine. Yeah.
Yeah, this I remember totally. This I remember from our previous discussion that I had a microfluidician around the table as well. So okay, so we have a bit of biology, biotech people, and a bit of microfluidician. So broad background. Okay, got it. So I'm not gonna go too deep on the biology side because you guys, I believe, are well aware that basically, so these RNA therapies, these new type of RNA therapies, they can be used mostly for three main applications. The first one is for infectious disease, to put together vaccines. We had the example of the COVID-19, but we have tons of vaccines under development at the moment regarding like flu, malaria, HIV, hepatitis, like tons of vaccines that can be used there. There is this RNA technology can also be used in the oncology space. So basically, people can you can use these to put together cancer treatment and personalized therapies, and also, and this is one of the biggest rising field that we see, which is really regarding gene therapy. Okay, gene therapies and gene editing, which are sort of like the new rising star recently, with many more applications open up in this field. We can come back to this if you're interested. And mostly, why is this new technology appealing? Mostly because it's much more simple than regular other technologies with deactivated viruses, for instance, and so it makes it cheaper, faster, and also with a broad potential. Now the big challenge of these technologies is that you need to introduce this fragile chain of RNA, of nucleic acid, within the cell to perform the function. And this is very tricky, and this is really where we bring our knowledge. Bear in mind as well, and I think this is an important data to keep in mind, that this is like the hottest, the most important topic for pharma and biotech companies, as nearly every one of them consider the RNA therapies either extremely or very important in the next few years. So this is definitely the next big thing in the space. All right. Now, when it comes to the different types of RNA and the cargo that we basically use, there are several ones that I would say, with these, it is 95% of our users that work with these three different types of RNA. Okay, the first one is everything that relates to mRNA, saRNA, or circular RNA, and the goal of these is basically to produce proteins, and so they're generally used a lot in vaccine development to enhance the immune response, and so to train the body against that, but also in gene therapy and cancer therapies. These things tend to be rather large, and so it's to be taken into account. The siRNA is the second thing that a lot of people also work with, and the first RNA lipid nanoparticle vaccines was actually by Alnylam in 2018. It was an siRNA LNP vaccines, and basically here you will use this siRNA to silence specific genes, and eventually this is just what you have to consider is that the size of this RNA is small. And the new thing that's coming up a lot recently is using LNPs to basically deliver all the CRISPR-Cas. So basically, bear in mind that here you got two things that you need to deliver. You need to deliver the guide RNA, which is around 20 bases, and the Cas9 and the mRNA that basically codes for the Cas9 protein. And you need to deliver the two together, and these can be used for gene editing mostly. All right, so a lot of application in the gene therapy space. All right, so these are, I would say, what 90% of our users are working with mostly, and this is, I would say, the wide majority of the cargo that you will see our users working with. All right. And now a quick overview of the different applications, but as I already mentioned, so infectious disease, so vaccines for those, genetic and rare disease, cancer vaccines, and personalized medicines. I insist on those because basically whenever you go to a university or you talk to the end users, basically if you know that they work, I don't know, in an oncology department, you know that there is a chance that basically they are interested in this technology. Or I don't know, if they work in a pharmaceutical department, they might also be interested in the technology, but it's just also to give you some insights on what are the different teams and different focus of the people working on it. Does that make sense so far for the very broad introduction to RNA therapies? I can dive way deeper, but I wanted to keep it very simple to start with. Is it all good? Do you have any questions on this? All right. Yes, I'll move forward then. Cool. So now, how do you deliver this RNA into the cells? Where there are several different methods that people can be using. The two historical methods that people have been using. The first one is was to use viral vectors, so basically lentiviruses and retroviruses, deactivated viruses. Okay, and basically the idea of those is simply to deactivate a virus and load it with your RNA and then deliver it to the cell. This is something that is it has very high cell internalization efficiency, so it goes very well into the cell. But the problem is that with this kind of delivery technology, you have safety concerns, you have a high toxicity, you cannot work with large DNA, and they are relatively difficult to load. So they work, but they're very, very far from ideal, and mostly they're also very, very difficult to produce. Okay. So the second methods people have been using is electroporation. So here the idea is, I don't know if you're familiar with electroporation. Are you? Maybe? Not really? Okay. So the idea of electroporation is very, very simple. You basically take your cells and you apply an electric field across the cells, and this will open up pores in the cell membrane. And basically these pores that are opening will allow for the RNA to get inside the cell. So you really just open doors in the cell thanks to an electric field. This is a very efficient method to use, but the problem with this kind of approach is mostly that you kill the cells, so there is a very high cytotoxicity. So the toxicity is very high. And also because, as you understand, you need the cells to do that. You cannot do directly into the body of the patients. So there is no in vivo, all right? You cannot do it within the body. You need to take the cells, take them out of the patient, do the electroporation, and put it back into the patient, all right? So it's what we call ex vivo approach. And so basically this is functional, but it is very, very, very expensive and makes the therapies completely very complex and very, very expensive. And so this is why people looked for more advanced carriers to be able to deliver RNA. And Eventually, people started working with regular polymeric or cationic-based nanoparticles, but these are very, very simple nanoparticles. But the problem is that the efficiency and the toxicity is usually low, and this is why people have developed this new type of nanoparticles, which you call lipid nanoparticles, which is now the gold standard for RNA delivery. And these are just sort of the best you can find to deliver RNA, because they have low toxicity. You can work with them in vivo. You can target them. You can customize them. The only hassle is that the process is slightly more complex than the regular nanoparticles. So this is why lipid nanoparticles is sort of what the pinnacle, I'll say, or what most people are using at the moment. All right. So this is the reason why LNPs. No questions on this? Is it clear? So one of the things on the sales side that we see a lot is that a lot of labs that used to work with viral delivery or that used to work with electroporation, they now are slowly switching to LNPs. Okay, and so typically we have a lot of labs that we know, they used to work with electroporation, and now they come to us and they say, oh, we have this first proof of concept with electroporation, but it's too expensive. We now want to switch to LNP. And this is a very, very standard case for users, all right? So very quickly, so a quick introduction to those lipid nanoparticles. So those lipid nanoparticles, as you can see, this is what they look like pretty much, okay? And basically you see that it's simply a shell made of lipids to get with a core, which is made of lipids as well, that is untrapping the RNA, okay? There is currently only 6 LNP-based drug approved as of 2024, but there is more than 1,000 drug under development at the moment, which are, and so that means there is a very, very big pipeline to feed, okay? And so really the goal of these lipid nanoparticles is to protect and deliver the RNA. Now, as you see, within the RNA, there is tons of different lipids with different colors. What are they more precisely? And this is, I believe, important to cover. So basically, these lipid nanoparticles, so these RNA lipid nanoparticles are made of 5 components. Okay, the first component is the RNA, okay, that we discussed before. It can be mRNA, siRNA, saRNA, all these kind of things. And then there is 4 other components that are critical to it. Mostly the most important ones are, so there is the helper lipid, which basically help with the stability of the nanoparticle. There is the ionizable, sorry, let me go for the least important one. The sterol lipid, which is mostly helping for the biocompatibility, okay, to be more compatible with cells. There is this PEG lipid, which is very important and present in most nanoparticles, because this is this long tail that you see here, this long stuff that you see there, which is this tail, and basically this is what is helping keeping the nanoparticle together, otherwise it would just sort of grow too big. And the most important part of the LNP is this ionizable lipid, okay, which basically can switch charge depending on the pH. And this is the key element of the nanoparticle, this ionizable cationic lipid, mostly because this is what combines with the LNP, as you see here. This is what is sort of combining with the RNA. Sorry, this is what is combining with the RNA and basically make this interaction unique. Do you have any questions on this, or is it clear? I can really go back in more detail on every single topic, but I just want to keep it simple for now, and so to answer your questions if you have any. Is it all good? All good, guys? All right, cool. Sounds good.
I have a question.
Please.
Yeah. What is the critical factor for formulating the LNP or liposome?
So many. Many. I think I have hold on. I think I have a slide on this after. I like to use this graph to see basically what are the interactions between what are the critical process parameters the critical quality attributes, and eventually the effect on the biology. Okay, so the stability, the toxicity, and so on. There is both the lipid composition that plays a role. Okay, so the lipid mix, the ratios of the different lipids, the concentration, what is the lipid you're using, and everything. Okay. There is the ratio of the RNA over the lipids. This also has a big impact. There is the composition of the buffer. There is everything that relates to downstream processing. So how do you purify and everything? There is the quality of the RNA that plays a role a lot. And there is also the formulation parameter. So eventually it's many parameters that are playing a role here. And this is one of the big problem. And yesterday, again, I was discussing it with a big French pharmaceutical company. And one of the big problem they have is that the process is very, very unstable because of the number of parameters at stake. You can even add external parameters like the temperature, for instance. Okay, all this basically is going to be affecting the nanoparticles. So it's very hard to answer your question because there is many different parameters. Does it answer your question? Is it all good?
Yeah.
Okay. Now, for what regards the LNP, more specifically, what is mostly studied is basically this ionizable cationic, this ionizable lipid. Okay, and this is where most of the research is currently being done. Okay. Now, if I have a quick introduction to the what do we look at when you, so the first thing you do before doing the biology is to have a look at the physical-chemical characterization of the LNP, and this is typically the different elements you look at first. So the first one, the first element you're going to be looking at is the composition of the LNP, okay? And you're going to be doing it using HPLC. I believe you guys are familiar with HPLCs, all right? The second thing you want to have a look at is what we call the colloidal, or the nanoparticle size and surface potential and everything. And this basically is done using DLS, okay, dynamic light scattering. I believe you guys might be familiar as well with this. The third thing most people want to have a look at is the encapsulation efficiency. Okay, so the percentage of drug that is encapsulated within the nanoparticles, so the percentage of RNA inside the nanoparticles. And basically you do this using fluorescence assay, so simply fluorescence. And something that is that we see more and more, it is the least looked at because it is very expensive. The last thing people would like to have a look at, because it's also important, is the morphology, and mostly the shape and the structure of the nanoparticle. And the problem is that you have to do this using cryo-TEM, okay? And this is very expensive. Okay, it's around 1 million USD, so very expensive. So this is basically what people look at when they talk about nanoparticles before going to the biology, so before doing the in vivo and the in vitro and then in vivo testing. All right. Do you have any questions on this? Because this is a very broad part as well. I don't know if you're familiar with DLS and everything, but it's just really what most people are looking at. So yeah, if I carry on, so maybe a quick overview of the overall RNA lipid nanoparticle vaccine development process, which I believe is very insightful. So typically the way this works is that there is a new virus that is discovered, people are doing the sequencing of the virus, then they design the corresponding RNA sequence, then they produce the RNA, they purify it, and you end up with your RNA, okay? And so once you have your RNA, basically hold on. Once you have your RNA, what you want to do is to put it inside your nanoparticles. And so what you're doing is that you're on the other side, you're taking your lipids and you pour them into a solvent. And what you do is you carry out a mixing step. This is where your nanoparticle is going to get formed, and basically this is the step we get involved in a lot. And then what you generally do is that you do a filtration of the process, you purify it, and you get your final drug product. So we are sort of the last step before purification in the process of the development of the RNA vaccines. All right. That was just to give you an outline of how this kind of RNA vaccines is being designed. So this is a quick recap of the approved RNA lipid nanoparticle technologies. At the moment, I'm gonna bypass this. There is no in Korea that I know of, so this is important. This is important to take into account. And now maybe this small introduction to the RNA lipid nanoparticle development step. And how does this kind of RNA lipid nanoparticle development vaccine development pipeline look like? So what is key to understand here, and this comes back to the questions that we just discussed before, is that there is many different parameters that are affecting the RNA lipid nanoparticle. Okay, we talked about the choice of the lipids, the concentration of the lipids, the ratios of the lipids, but you also talk about the choice of buffer. You also talk about the choice of the RNA sequence. You talk about the quality of the RNA. You talk about the quality of the lipids. And so many different parameters to test. And there is no predictive model at the moment for it. So the only way to do it is to use screening. Okay, so the first step of a development process is to carry out screening, meaning to testing many different type of RNA lipid nanoparticle together, okay, and finding which is best, all right, for them. And so the big challenge here is the following. The big challenge is that RNA is very expensive, okay? You're talking about roughly around 2,000 USD per milligrams, which is very expensive. And so if you want to be able to do low volume, if you want to be able to test hundreds or thousands of formulation, basically what you need to do is to work with the smallest amount of liquid. So because otherwise it will get insanely expensive, very expensive. So this is why the first development step here generally is screening, so testing tens, hundreds, thousands of different formulations. All right. Once you've done this, what we see people doing is switching to preclinical testing, where eventually, so they identify the most promising RNA lipid nanoparticles, and they will then test it in vivo on mouse, and eventually going all the way up to monkeys or pigs. And basically here you need typically more volumes in the milliliter range because you want to inject many different mouse. And so basically what happens is that this is what covers the research and development steps. And then you switch to the clinical testing where you test it on human and then manufacturing. At the moment, and just for you to know, Tamara is really well positioned for the R&D stages. So only for the screening and preclinical testing. Okay, so this is what I'm mostly going to be discussing today for the R&D side. We will next year come with a solution for the scale up. Okay, but at the moment, we are only focusing on research and development, and so on the screening step and on the preclinical testing step. All right, this is really where our market is at the moment. All right, and so who are the users we work with? So the academia, so the academic labs, the biotech and pharma, but their research and development department mostly. All right. So this is mostly the people we work with. Now a very quick opening to what we're doing. So right now, we've mostly been discussing about LNP for RNA and DNA and delivery. And this is, this is something 80% of our users work with. It works great for all applications. We never had any trouble. You can work with pretty much any payload. We have a lot of data and so on. Okay, so this is what I'll say 80% of our users work with. Then there is another 10% of the users that you see here in the middle that are doing liposomes, PLGA, or peptides nanoparticle. Generally, we have great results with these. We have a lot of data, basically, so this is working great, but this is a field we have less data on. But this is definitely doable. Eventually, people can also do pretty much any other type of organic nanoparticle. So polymer, lipid, peptides, hybrid nanoparticles using our systems. Basically, the question you need to ask people when you don't know is, can you make the nanoparticle using hand mixing? If so, we can do it better, all right? So basically what we can make is any nanoparticle that can be made by hand mixing. Tamara can do it too, with better control, reproducibility, and scalability and encapsulation efficiency. This is basically when you meet someone and you don't know what to do, and you know, if they don't do RNA LNPs or they don't do very simple liposomes, if they can do it by hand mixing, you can do it, but better. All right, this is kind of the idea. Does it make sense to you on this? Okay, all good? Okay.
Okay, understand. Just a moment. 신 과장. 이게 지금 그거 하는데 지금 10분밖에 안 남았는데.
네, 이게 저 시간 제한 없는 줄 알았는데 시간 제한이 있네요, 이거.
그래, 그럼 어떻게 할 거야?
나갔다 다시 들어와야 될 것 같아요. 좀
그래?
이따. 네.
지금 4분 남았어.
그래?
어디 10분까지 나오고.
아.
내가
Okay, can I interrupt you for a second?
Sure.
Yeah,
Yep.
I didn't know there is some time limits because I just go in the website and there was the teams there was no time limit, so we didn't purchase the membership. So if possible, can we enter again for this meeting room? Just leave for a while and enter this meeting room again.
Yeah, sure. I can
Yeah.
I can also send you a link. I have no problem with Teams, so we can have very long meetings.
Mm,
I'm not sure I understood your point.
we have a time limit. We just left 3 minutes for this meeting room, so
Okay,
We
all right,
have
sure,
to-
no problem.
go out
No
and
problem.
enter this room again.
That sounds good. Yeah, go ahead, please.
Okay, thank you.
I'll wait for you.
Yeah.
그렇지. 안된것 같은데 57분, 아직 57분이니까 안 됐어요.
Do I need to leave as well?
나가기 옆에고 다른 건
모임 종료해야겠네.
예.